Dear teachers and my dear students, myself Dr. P.K. Verma. Today in the 10th session of Human Physiology, I shall discuss excretory system. Thereafter in the 11th session, I shall discuss the mechanism of urine formation. Excretion is the process of elimination of nitrogenous waste product to maintain the internal chemical milieu. Usually, people say that elimination of anything is excretion. They also say about the sweating and spitting as a type of excretion. Sometimes the term is used also for the plants. But in true sense, excretion is the process of elimination of nitrogenous waste products in the animals. These waste products or most of these products are used by the plant. So truly in the plants there is no excretion. Ammonia is regularly produced by deamination of amino acids. It is one of the worst products, that being highly soluble, diffusible and alkaline neurotoxic chemical, needs immediate elimination or converted into urea to reduce toxicity. Excretory system is mainly concerned with the removal of ammonia and other nitrogenous wastes. In different animals, there are different excretory products. For example, the ammonia can directly be released out of the cell surface in the unicellular organisms such as amoeba, chlamydomonas, etc. It is also released from the body surface of sponges and hydra. In the tadpole larvae of amphibians and the fishes, ammonia is released from the surface of gills. However, in the adult amphibians and mammals, urea is excretory metabolite. In terrestrial insects, reptiles, birds, and egg-laying mammals, uric acid is the chief metabolite. In birds, ornithine as well as guanine also are excreted. The spiders also excrete guanine. However, in the marine fishes, urea is retained inside the blood and body fluid to make higher osmotic pressure equivalent to the osmotic pressure of seawater. That is, they need to maintain their body fluid and blood isosmotic with marine water, and therefore they retain urea, and in place of urea, trimethyl amine oxide is excreted. The animals that excrete ammonia are called ammoniotelic. Those excreting urea are ureotelic, and those excreting uric acid are called uricotelic. In the embryo, there is allantois serving as urinary bladder. They excrete allantoic acid. In frog, human beings and other mammals, urea is synthesized in liver by arginine ornithine urea cycle or Krebs-Henselt cycle. The urea passes from liver into circulation through hepatic vein and filtered by kidney. This is the diagram showing the pathway of urea synthesis. At first, carbon dioxide and ammonia combine together along with phosphate, forming carbamoyl phosphate. Later on, the ammonia group is transferred to ornithine and therefore citrulline is formed. Aspartic acid also contributes amine group to citrulline and form arginine. From arginine, urea is released and again ornithine is recycled. In urea, there are two amino groups. One is directly derived from the free ammonia and another is derived from aspartic acid. So if there is poor amount of aspartic acid in our diet or it is not produced inside the cells, the urea synthesis is affected. In the bottom of the diagram, the full names of enzymes used in the urea synthesis has been written and their abbreviations such as CPS, OTS, ASS, ASL have been used in the diagram. In the animal kingdom, excretory system evolved gradually and therefore in different phyla, different types of organs of excretion were formed. As I have discussed, in the protozoa, ammonia is released from cell surface and in sponges and coelentrates, it is released directly from the body surface because the inner and outer cell layers are directly in contact of water. However, in the flatworms, flame cells are formed and in roundworms, rennet cells are formed. In the roundworms, 3 or 4 rennet cells join together, become syncytium and form H-shaped excretory system. In the annelids, higher type of organization developed and they formed nephridia. In earthworm, it is present in large number, but in Hirudinaria there are counted number of nephridia in different species. In the arthropoda, malpighian tubules are present, especially in the insects, but in crustaceans, special coxal and green glands are formed. In mollusca also green gland is present, but in protochordata, solenocytes are formed that look very much similar to the flame cells. In the vertebrates, however, the well-organized nephrons and kidney has been developed. Now, let us study about the human excretory system. In humans, a pair of kidneys have been developed in the abdominal region from the mesodermal somites. from the same somite, adrenal gland, and urinogenital system, that is ovaries and testes, also have been developed. In fishes and amphibians, the kidneys are mesonephric, but in reptiles, birds, and mammals, these are metanephric kidneys. There is a pair of kidneys in the two sides of dorsal aorta in the middle of abdomen in humans. These are dark red in color due to very high blood supply directly from dorsal aorta through a pair of renal arteries. The blood returns through a pair of renal veins into posterior vein. Each kidney is bean-shaped with an inner depressed part known as hilus. To this part, renal arteries and renal veins are attached and the ureters come out. See the full structure of kidneys and arrangement of the arteries and veins to kidney. In the right kidney, only the attachment of arteries, veins and the ureter is shown, but in left kidney, you can see branches formed of veins in this diagram. In fact, the vein is dorsally to artery. A kidney measures 13 cm by 6 cm in size, the left being somewhat larger than the right kidney. The two kidneys together weigh about 300 grams in the male and about 250 grams in female. In the longitudinal section of kidneys through lateral side, that is known as sagittal section, its internal anatomy can be studied in details. Malpighi, who is honored as father of microscopic anatomy, at first described internal structure of kidneys. He observed two major parts, two distinct parts inside kidney. The outer is called cortex and inner is medulla. In the cortex, he found granular structures that were named as Malpighian corpuscles, but later on it was found that it is the glomerulus covered with the Bowman's capsule. In the medulla, there are fine striations radially arranged, and these are nothing but the long tubular part of the nephrons that form the organ of excretion or the units of excretion. See in this diagram, the outer part is cortex, inner part is medulla. In the medulla, the urine collecting ducts form minor calyx and 2 or 3 such minor calyces join together to form major calyces. These minor and major calyces ultimately open in a common space known as renal pelvis. From the renal pelvis, the ureters come out. In between the minor and major calyces, you can see the connective tissue is present along with some fat. This part is called column of Bertini. The latter anatomical studies show that there are about 200,000 tubular filtration units in each kidney. These are known as uriniferous tubules or nephrons. These nephrons are arranged radially within the kidneys and form urine. These tubules extend from cortex to medulla, but being very long, they form a loop of Henle in the medulla and in both the limbs of this loop. Coiled tubular structures are seen behind the neck of Bowman's capsule as well as between ascending limb and the collecting duct. These are known as proximal and distal convoluted tubules. See this structure of nephron. Here the apical part of nephron forms a cup-shaped structure into which afferent renal artery enters, forms the capillary network, bunch of capillaries known as glomerulus, and then all glomerular capillaries join together to form the efferent arteriole. Later on, the efferent arteriole forms peritubular capillary surrounding the two arms of the loop of Henle, that is the descending and ascending limb. In both the limbs, the lower part is thinner than the upper, thicker parts. In between the neck and descending limb, there is proximal convoluted tubule. See, this part is proximal convoluted tubule and this one is the descending limb. When it moves outward towards the cortex, it is called ascending limb, and again there is a convoluted part known as distal convoluted tubule. Ultimately, a collecting duct is formed. The formation of collecting ducts, their joining and rejoining, results into thickening of the collecting duct gradually, so the lower part is wider than upper part and therefore the lower part is called pyramid. This arrangement of tubular structure is called pyramid. You can compare it with the branching of trees, that is an arborized system, tree-like system into which the stem is thicker than the branches, and from each branch so many finer branches appear. Similar is the arrangement of the nephron with collecting ducts. In the cortex, they have a cup-shaped Bowman's capsule made of two walls. The inner wall is made of thin layer of cells, but the outer wall has cuboid or columnar cells. Behind the capsule there is a short neck followed by proximal convoluted tubule. The distal part of these tubules again return from cortex to medulla and form the collecting duct. Thus, a single tubule forms three parallel branches, parallel arms in the medulla. One is descending limb, second is ascending limb, and the third becomes collecting duct. The terminal part of collecting duct becomes thinner, that is known as duct of Bellini, and they join together to form an arborized system of collecting ducts that is formed in different parts of kidney. All these pyramids form minor calyces and then major calyces and finally open in a common space known as pelvis, from which ureter carry out the urine. The connective tissue from cortex to medulla is continued in between these pyramids that forms the column of Bertini. Now again you can see the arrangement of nephrons. The diagram is very clear. From Bowman's capsule, proximal convoluted tubule, then descending limb, thinner part of descending limb, thinner part of ascending limb, again thicker part of ascending limb, distal convoluted tubule, and finally collecting duct is formed. In different part of nephron, the cells have different structures. The inner wall of Bowman's capsule. Here is the Bowman's capsule. It is the inner wall. Is very thin, made of squamous epithelium, similar to the wall of the blood capillaries. But thereafter in the neck region, it has so many microvilli or brush border. So after filtration, the useful substances will be easily absorbed by these brush borders because the surface area increases very much by formation of these microvilli. Now let us see how the blood is supplied inside kidney. The renal arteries divide into a large number of interlobar arteries. You have seen the diagram of sagittal section of kidney. There were different parts known as lobe. So the branches passing into each lobe is interlobar artery. And then in between cortex and medulla, they form the arcuate artery. It runs parallel to the surface, and thereafter the branches passing into cortex form interlobular artery. Interlobar forms arcuate artery. Arcuate forms interlobular arteries at right angle to the surface, from which afferent arteries are formed. See how the arrangement of vein and arteries are there inside kidney. This is the vein. It collects blood from nephrons. So these branches around the lobes of kidney are interlobular branches. They form arcuate vein, then they join together to form interlobular vein. These interlobular veins again join together and forms the interlobar veins, and finally they come out as renal vein. Similarly, the renal artery divides into a number of interlobar arteries, and these interlobar arteries form the interlobular arteries, arcuate arteries, afferent renal arteries, efferent renal arteries, and peritubular capillaries. Each afferent artery enters into Bowman's capsule and a bunch of capillaries is formed. It is known as glomerulus. The glomerulus and Bowman's capsule together form Malpighian corpuscles, and these are the sites of filtration of blood plasma. The back journey of blood has slightly different path from the peritubular capillaries. Interlobular veins, arcuate veins, interlobar veins are formed that finally join the renal vein. The tunica media of afferent arteriole is made of highly granular cells known as juxtaglomerular cells. These are very important physiologically. When there is low sodium inside blood, these cells secrete enzyme renin that helps in absorption of sodium from the descending limb, ascending limb, and collecting ducts. Renin helps in absorption of sodium by active transport, but it also allows discharge of potassium. Now let us see how the ureters carry urine from kidney to urinary bladder. In humans, the ureters are usually 25 to 30 centimeter long, that is 10 to 12 inches in length. These are muscular ducts that propel urine from kidney to urinary bladder. The ureters run back on the lateral walls of pelvis. They then curve medially to enter the bladder through the backside, running within the wall of bladder for a few centimeters. In the females, the ureters pass through the mesometrium of uterus on the way to the bladder. The backflow of urine is prevented by valve in between ureter and the urinary bladder. This valve is known as ureterovesical valve. When this valve is closed, urine cannot pass back to kidney. It is unidirectional, kidney to urinary bladder, not from urinary bladder to kidney. The urine is carried into urinary bladder by a peristaltic movement or waves of ureters. These waves originate in a pacemaker at the renal end of ureters, that is near kidney. The wave occurs every 10 seconds or so and travels down at 2 or 3 centimeters per second. Thus, each wave sends only a small spurt of urine into the urinary bladder. It does not flow continuously. A small amount of urine is taken from kidney and passed to urinary bladder every 10 seconds. The ureteric muscles are fairly powerful, since if the ureters are obstructed, the ureteric pressure may rise to 50 millimeter of mercury. Now, see the structure and function of urinary bladder. The urinary bladder is the organ that collects waste excreted by the kidneys prior to disposal by urination. It is a hollow muscular and distensible organ and sits on the pelvic floor. Urine enters the bladder via the ureters and exits via the urethra. Embryologically, the bladder is derived from urogenital sinus and it is initially continuous with the allantois. allantois is found in embryo. In human males, the base of bladder lies between the rectum and the pubic symphysis. It is superior to prostate gland and separated from the rectum by retrovesical excavation. In females, the bladder sits inferior to the uterus and anterior to vagina. It is separated from the uterus by the retrovesical excavation. So the opening differs in the male and female. In infants and young children, the urinary bladder is in the abdomen even when empty. The urinary bladder is lined by transitional epithelium. That is, the number of cells in the layer changes from 5 to 6 layers to only 2 to 3 layers when it becomes distended. Their shapes change easily when the bladder is distended. The epithelium is firmly adhered to the wall of smooth muscle fibers surrounding the inner epithelium. At the base of the bladder, the smooth muscle fibers pass around the urethral opening, forming internal sphincter or sphincter vesicae. These are supplied by hypogastric nerve and the pelvic nerve. The external sphincter surrounding urethra is under voluntary control through the peduncle nerves, which are somatic nerves. Opening of the urinary bladder into urethra is therefore under voluntary control except the extreme situations. In normal circumstances, the person becomes aware of fullness of bladder when about 150 or 160 milliliter urine is stored. The bladder is allowed to fill until it contains nearly 300 milliliter urine. After the bladder becomes full, the pressure receptors in its wall become activated. Now, emptying of bladder occurs by impulse passing down the pelvic nerves, which causes the detrusor muscles to contract and open the internal sphincter. The external sphincter relaxes when the intravesical pressure is above a threshold value from 20 to 40 centimeter of water. Although it is a striated muscle, it cannot be relaxed voluntarily. It may be consciously contracted. If forced to micturate does not produce a relaxation of the external sphincter. Inhibition of sphincter occurs only when the detrusor muscles contract. The internal sphincter relaxing a little before external muscles relax. The stream of urine can, however, be stopped by a voluntary contraction of external sphincter, though it is very difficult. If more urine is allowed to accumulate, a sense of urgency arises, and when the bladder contains about 600 ml urine, the contraction of blood vessels become so powerful that the pressure is raised to 100 centimeter of water and it causes pain. It is a very harmful condition. It can be restrained voluntarily for a short time only. Thereafter, the discomfort becomes inevitable and urination is compulsion. So you have studied all about the structure of kidneys, how the urine is collected into urinary bladder and how the urine is discharged. In the next session, I shall discuss how urine is formed, how the blood plasma is filtered and converted into a highly condensed urine. Thank you. Thank you very much.